Okay, hello to all of you. Let's discuss chapter number 13, and this is the last chapter of the course. We are not doing the last unit. Um, in the chapter number 13, we are going to talk about the cytoskeleton, and in this, we are going to talk about the cytoskeleton filaments, which are made of proteins. So, basically, first of all, what does the word cytoskeleton means? It's the skeleton of the cell. So just the way the skeleton of our body helps us in movement and it helps in movement and gives us shape. Similarly, the cytoskeleton, which is the skeleton of the cell, gives the cell its shape. It gives basically helps the cell in its movement. So and basically the cytoskeleton is made of protein filaments. So this is just a picture of the cytoskeleton of the cell, and it is made of basically these are the major structural elements of the cytoskeleton. They are microtubules, microfilaments, and intermediate filaments, and these are made of proteins, and we are going to discuss about them in detail. The microtubules are composed of tubulin subunits. Microfilaments, they are composed of actin subunits, and intermediate filaments, they are variable in the composition. So this table basically shows you the differences between the three cytoskeleton um, subunits. Sorry, the cytoskeleton filaments. So these are microtubules, these are microfilaments, and these are intermediate filaments. So you can see the shape of the microtubules. So they are like hollow tubes. So they are like these. They are hollow tubes with a wall consisting of typically of 13 microfilaments. So these are hollow tubes consisting of typically 13 microfilaments. So this is not sorry microfilament, it's protofilament. So this is one protofilament, and they are made of proteins like tubulin, and there are two kinds of tubulin, alpha and the beta tubulin, and they basically heterodimerize. Alpha and beta tubulins, they heterodimerize, and the microtubules, they are made of these beta tubulins. And there are two kinds of microfilaments: cytosolic microfilaments and axonal microfilaments. So, axonal microfilaments, they are involved in the cell motility, and cytosolic microtubules, they are actually they are involved in the chromosome movement during the mitosis and meiosis, and also they are involved in the movement of the organelles. So the next cytoskeleton filament is microfilaments, and these microfilaments are made of a protein called actin, and the microfilaments are basically made of two twined of the F actin. So you can see that these are the actin proteins. So these are each is called the G actin monomer, G for globular, and they polymerize and they form the filaments. So these are the two intertwined chains of the F actin, and every F actin, so this is F means filament, and it is made of all these G actins. Um, Basically, the microfilaments, they are involved in muscle contraction, they are involved in cytoplasmic streaming, and also maintenance of animal cell shape, intracellular transport, and trafficking. And the last one is intermediate filaments, and these intermediate filaments, they are basically made of 8 protofilaments joined end to end with the staggered overlaps, and they are made of 6 different classes of the proteins, and they are involved in the formation of nuclear lamina, and they are also involved in strengthening of the nerve cell axons. So these are some of the important functions and some of the important differences between these three different cytoskeleton filaments. So let's go to microfilaments. So microfilaments are the largest structural element of the cytoskeleton, and they are involved. So as I said, microtubules, sorry, we are talking about microtubules, they are the largest. And microtubules, as you know, there are two different kinds. They are cytoplasmic microtubules and they are axonal microtubules. So cytoplasmic microtubules, they are responsible for the following functions. They are responsible for maintaining axons, formation of mitotic and meiotic spindles, maintaining or altering cell shape, placement of movement of vesicles. Axonal microtubules, they are found in the structures like cilia and flagellum, and also the basal bodies from which the cilia, to which the cilia and flagella attach. Microtubules, they are basically straight hollow cylinders, as I just showed you. They consist of usually 13 longitudinal arrays of the polymers called protofilaments. And the basic subunit of a protofilament, the basic subunit of a protofilament is a heterodimer of tubulin, one alpha tubulin, another beta tubulin. And these bind non-covalently to form alpha beta heterodimer. So you can see this is one protofilament, this is protofilament, alpha beta, and there are different types of microtubules. They can be singlets, they can be doublets, or they can be triplets. So the alpha and beta subunits have nearly identical three-dimensional structure, but only 40% amino acid identity, and each of them has N-terminal, which is a GTP binding domain, and central domain to which called cycline, which is a kind of a drug. It binds, and a C-terminal domain that interacts with the microtubule associated proteins. And all dimers in the microtubule are oriented in the same way. So basically there are two ends of the microtubules. The growing end is called the plus end and the other end is called the minus end. So this is the polarity of the microtubules. So the growing end is called the plus end and the other end is called the minus end. So cytoplasmic microtubules are usually simple tubes or singlet microtubules with 13 protofilaments. Axonal microtubules are microtubules. So doublets and triplets contain one 13 protofilament tubule and one or two additional incomplete rings like B and C tubules of 10 or 11 protofilaments. So microtubules, they are actually formed by the reversible polymerization of the tubulin dimers in the presence of GTP and magnesium. So these alpha and beta tubulin, they form the heterodimers, and these heterodimers, they then form the oligomers. Okay, and oligomers, they serve as seeds from which the new microtubules they grow. So dimers aggregate into oligomers which serve as seeds from which new microtubules they grow. This process is called nucleation, and the addition of more subunits at either end is called the elongation. So microtubule formation, first of all, is slow, and the process of, because in the initial phase it's a process of nucleation, that's a slow process, and this period is called the lag phase. And then after that, after the lag phase, is elongation phase, which is much faster. So when the mass of the microtubule reaches a point where the amount of free tubulin is diminished or it is reduced, the assembly is balanced by disassembly, and this is known as the plateau phase. So this slide shows you everything. So basically you can see these are the heterodimers. So this is a heterodimer of alpha and beta tubulin. So you can see this is a dimer, this is a dimer. First of all, they form the oligomers. Okay, so these oligomers, as it's written here, the dimers aggregate into oligomers which serve as seeds from which new microtubules grow. So you can see here these are the heterodimers, and they are forming the oligomers, and from the oligomers these they act as seeds, and these microtubules they can be formed like this. So as the microtubules, they have a polarity. The end of the microtubule which is growing is called the plus end, and the other is called the minus end. So you can see how these dimers, they are getting attached on this side. Okay, so first of all, this phase, when all the nucleation is taking place, all these are forming these oligomers. So this phase is called lag phase, and this is little slow phase. And after that is elongation phase, which is quite fast. And after that reaches the plateau phase. So in the plateau phase, basically the assembly is balanced by disassembly. Okay, because the amount of the free tubulin decreases, and as a result of that, at that point the assembly is balanced by disassembly, and this phase is called the plateau phase. So you can see here the amount of free tubulins which are available at this phase, they decrease, and as a result of that, what happens is, whatever the tubulins are being assembled here, the same amount of tubulins are getting disassembled from here. So this is called the plateau phase. So microtubule assembly in vitro depends on the concentration of tubulin dimers. So the tubulin concentration at which microtubule assembly is exactly balanced by disassembly is called the critical concentration. So the concentration of the tubulins at which microtubule assembly is exactly balanced by disassembly is called the critical concentration. So microtubules grow when the tubulin concentration exceeds the critical concentration. So critical concentration is that concentration at which the microtubule assembly is exactly balanced by the disassembly. That is called the critical concentration. So microtubules will grow when the tubulin concentration which is available exceeds the critical concentration. Otherwise, at the critical concentration, the assembly will balance the disassembly. Okay, so we already know the rapidly growing microtubule end is called the plus end, and the other end is called the minus end. So you can see here, this is showing the basal body. So in this side is the plus end, and this is the minus end. So the plus and the minus ends, the plus and the minus ends of the microtubules, they have different critical concentrations. So if the free tubulin concentration is above the critical concentration for the plus end but below that of the minus, then treadmilling will occur. So what is treadmilling? It is the addition of the subunits at the plus end and the removal from the minus end. So basically, so you know that the microtubules, they have two ends, and the minus end. If the free tubulin concentration which is available is above the critical concentration for the plus end, so if the tubulin concentration which is available is above the critical concentration of the plus end but below that of the minus end, then what will happen? The process of treadmilling will occur, which means that there are two ends of the microtubules, plus end and the minus end. If the concentration of the two ends, they have different critical concentrations. So the amount of tubulins, the free tubulins which are available, the tubulin dimers which are available, if their concentration is more than the critical concentration of the plus end, then the assembly will take place on the plus end. But because the tubulin concentration available is less than the critical concentration of the minus end, disassembly is going to take place from the minus end. So if that happens, then a process called treadmilling occur. Now what is treadmilling? Addition of the subunits at the plus end and removal from the minus end. So you can see here in this situation, the tubulins will be added here, but they will be removed from here. So you can see here that whatever is, so basically It's happening because the two ends, this is a plus end, this is a minus end. The concentration of the tubulins which is present is above the critical concentration of this end, so they will be added here. But the concentration of the tubulins which is available is less than the concentration on this side, is less than the critical concentration of this end. So disassembly will take place on this side. So on this side assembly, this side disassembly. This process is called treadmilling, and this is basically showing that whatever is being attached on this side is actually like this. We started from here, so how they keep on moving, because on this side continuous assembly is taking place, and on this side continuous disassembly is taking place. So eventually this part which is they're showing just with blue, it will be also disassembled. This process is called treadmilling. And now we are going to just talk about some of the drugs which can affect the assembly and the stability of microtubules. Colchicine, it binds to the tubulin monomers and it inhibits their assembly into microtubules. This is vinblastine and vincristin. They are also related products. Then this drug nocodazole also inhibits the microtubule assembly. And these drugs, because they affect the microtubule assembly, they inhibit the microtubule assembly. So they can be because microtubules are very important in the process of cell division. In the cell division, the mitotic spindle is formed, and mitotic spindle is involved in the separation of the chromosomes. So microtubules are involved in the process of mitosis. So because these drugs, they affect the formation of microtubules, they can be used as anti-mitotic drugs, and they can be used for cancer treatment. Now the Taxol, you have already studied docetaxel in the lab, and basically docetaxel or the Taxols, they basically prevent the depolymerization of the microtubules. So this is a table, you can go over it. Each tubulin heterodimer binds two GTP molecules. So you know that the tubulin heterodimer has two kinds of proteins, alpha tubulin, another is a beta tubulin. And alpha tubulin and the beta tubulin, to both of them GTP is bound on them, but GTP bound to the beta subunit is hydrolyzed to GDP after the heterodimer is added to the microtubules. So as the microtubules are getting assembled and these heterodimers are added to the microtubule, the beta subunit actually, both the alpha and the beta subunit, they have GTP bound to them, but the GTP which is bound to the beta subunit is hydrolyzed to GDP. So the growing microtubules, so they have GTP at the plus end, and shrinking microtubules, they have GDP. So the GTP cap at the plus end prevents subunit removal. So you can see that this is the growing site, because on this side is the GDP. So this is called the GTP cap. So if there is a high concentration of GTP, tubulin concentration is high, then the GTP tubulins will be added on this side. Okay, and in the conditions when the GTP tubulin concentration becomes low, then the GTP will be hydrolyzed on this side, and the GTP cap will be depleted. So if the GTP tubulin is high, as I said, it is added to a microtubule quickly, creating a large GTP tubulin cap. If the concentration falls, the rate of tubulin addition decreases. At a sufficiently low GTP tubulin, the rate of GTP hydrolysis exceeds the rate of subunit addition and the cap shrinks. So that's all what we just talked to here. If the GTP cap disappears altogether, the microtubule becomes unstable and loss of GDP bound subunit is favored. Individual microtubules can go through periods of growth and shrinkage. A switch from growth to shrinkage is called the microtubule catastrophe, and sudden switch back is called the microtubule rescue. So you can see that this is the two ends of the microtubules, plus and the minus end, and you can see here the plus end is the growth is taking place. So at this point, when the growth is taking place, the GTP tubulin is added, and when the GTP is hydrolyzed, then the microtubules they depolymerize, and this is called microtubule catastrophe. And if the growth resumes back, this is called microtubule rescue. Okay, these microtubules they basically originate from microtubule organizing center, and this microtubule organizing center is also called the it's basically the centrosome. And this centrosome, so this is a region outside the nucleus, and this region has actually two centrioles which are like rod like structures, and these centrioles they are made of microtubules, and these centrioles are surrounded by a pericentriolar material. So this region called centrosome, which has two centrioles, this region is called microtubule organizing center. And this basically, you can just go over it. It basically talks about the structure of the centrioles, those rod shaped structures which are sitting perpendicular to each other. They are centrioles. They are formed by 9 pair of triplet microtubules, and you can see here. So they are and centrosomes have ring-shaped protein complexes in them, and these are called gamma tubulin. So gamma tubulin is found only in the centrosome. So gamma tubulin ring complexes nucleate the assembly of new microtubules away from the centrosomes. So loss of these tubulin ring complexes prevents a cell from nucleating microtubules. So you can see that this is the centrosome, and this is the gamma tubulin ring, and you can see these are this region is called centrosome region, and these are the centrioles, and this region is called the microtubule organizing center, because from here microtubules originate, and this side which is attached to the centrosome, this is the minus end, and the other side, the other end is the plus end, and this is the growing end of the microtubules. Okay, so basically this is showing us, this slide is showing us actually how from the centrosomes these microtubules they originate. Okay, so it's showing you this is the interface, this is the nucleus, and these centrosomes, this centrosome is present actually outside the nucleus, and from here these microtubules they originate, and you can see that there are actually two centrosomes, because in the interface there's duplication of all the organelles, and these centrosomes they also actually duplicate, so they start moving apart, and you can see that they reach two different poles, and so the side of the microtubule which is attached to this centrosomes, that's the negative, that's the minus end, and the growing end is the positive end. So you can see that these are the centrosomes, and this is the microtubule organizing center, and how the microtubules, they originate from them and how they form the mitotic spindle. Yeah, now we are going to talk about some of the microtubule binding proteins. So some of the proteins are called tau proteins, then MAP2 proteins. So you need to know what is the function. So MAP means microtubule um, associated protein. Tau causes microtubules to form tight bundles in the axons. MAP2 protein promotes the formation of looser bundles in the dendrites. So proteins like tau and MAP2, they have two regions. One region binds to the microtubule wall, and the other part of the protein extends at the right angle to the microtubule to allow the interaction with other proteins. So you can see here that this is a tau protein, that how it is, this part of the protein is binding with the wall of the microtubule, and this part is free so that other proteins can bind to this protein. So this is a microtubule associated protein, and these proteins, they have different functions, and this is the list of some of the more proteins. So the microtubules can be stabilized by proteins that capture and protect the growing placents, and these are called tip proteins. So tip means tubulin interacting proteins, and there are proteins like proteins such as EB1. These are called end binding proteins. They decrease the likelihood that microtubules will undergo catastrophic subunit loss. And these are some of the, you can go over the names, and these are the microtubule destabilizing or severing proteins. So you can go over these names, and they're important. So the next cytoskeleton element we are going to discuss, the filament we are going to discuss, is the microfilament, and they are the smallest of the cytoskeleton filaments. The basic role is in muscle contraction, in the cytoplasmic streaming, amoeboid movements, and cell migration. And they are made of a protein called actin, and this protein, once this protein is synthesized, it folds into a globular shaped molecule that can bind ATP or ADP. So this protein, so this actin protein, when it folds into globular shape, uh, they are called G-actin globular. They're called G-actin, or they are called globular actin. And G-actin molecule polymerizes to form microfilaments F-actin. Now these actins can be classified, can be broadly divided into muscle specific actins, alpha actins, and non-muscle actins, beta and the gamma actins. Now these G-actin monomers, they can polymerize reversibly into filaments with a lag phase and elongation phase, similarly to the tubulin assembly. Just the way we saw how the polymerization of the microtubules takes place, it's quite similar for the microfilaments, which are basically made by the polymerization of the G-actin monomers. The F-actin filaments are composed of two linear strands of the polymerized G-actin wound into a helix. So, so you can see here that this is, as you know, that they are like two, basically they are composed of two linear strands of polymerized G-actin. So what we are going to see in the slide is F-actin filaments are composed of two linear strands of polymerized G-actin wound into a helix. So you can see here that this is all G-actin, okay, this is all G-actin, and how they have polymerized and they have formed the F-actin, and how their two chains are wound around each other. So again, microfilaments also have a polarity, and it is reflected in a more rapid addition or loss of G actin at the plus end than the minus end. After the G actin monomers assemble into the microfilament, the ATP bound to them is slowly hydrolyzed. So you can see that all these G actins, they are polymerized. So you can see that on this side you have G actins with ATP on them, and on this side you have all the G actins with ADP on them. So the growing microfilament ends, they have ATP actin, whereas most of the microfilament is composed of ADP actin. And this is some of the specific drugs which affect the polymerization of the microfilaments. Now let's see in some of the structures where filaments are involved. So cells that adhere tightly to the underlying substratum, they have organized bundles called stress fibers. Okay, and the cell cortex just beneath the plasma membrane, they also have actin cross-linked into gel of microfilaments. So this is showing you basically the cell. So when the cell move, they form projections like this. They are called philopodia and the lamellopodia. So lamellopodia, philopodia, and this is called the cell cortex. Okay. And this is showing you all the arrangement of these actin microfilaments, how they are arranged. So in different different for projections like philopodia and lamellopodia, they have a different arrangement of the these actin filaments. So it is like in the lamellopodia, it is like branched. In the philopodia, they're parallel, and like this is for the cell cortex, they're like a gel, and for the stress fibers, they are like contractile bundles. So cells that crawl, so lamellopodia and philopodia, they are basically projections that help in the movement of the cell. So the cells that crawl have lamellopodia and philopodia at the leading edge, allowing them to move along the surface. Lamellopodia have branched network of the actin. So this is lamellopodia. Okay, this is the lamellopodia. These are called lamellopodia, and they have the branched network. And the philopodia at the leading edge filaments form highly oriented polarized cables with the plus end towards the tip of the protrusion. And now we are going to talk about some of the proteins which are associated with the microfilaments. And here shows a protein called thymosin, and this is a monomer sequestering protein. So you can see all these are the actin monomers, and these proteins like thymosin, they basically bind to them and they sequester them. They sequester these proteins bind to these actin monomers and they just sequester them. And this is actin polymerizing protein called formalin. This formin. It helps in the actin polymerization. Then this is filament severing proteins like gelsolin, uh, and this is filament capping proteins, CAPZ, filament cross-linking proteins, filament, then filament bundling proteins like alpha actinin and fibrillin. Okay, so this is basically all the information I just gave you. You can go over them. So they are all proteins which interact with the actin filaments. Okay, the last one is intermediate filaments. Um, they are very much abundant in animal cells, and the abundant intermediate filament is keratin, and it's an important component of structures that grow from skin in the animals, and they are the most stable and least soluble components of the cytoskeleton. So they can be classified into 6 classes. Class 1 are called acidic keratins, called basic keratins. Class 3 includes vimentin, which is present in the connective tissue, then desmin in the muscle cells, then glial fibrillary acidic proteins. Class 4 includes neurofilament proteins found in the neurofilaments of the nerve cells. Class 5 includes nuclear lamins A, B, and C that form a network along the inner surface of the nuclear membrane. And then class 6 has nestin that makes up the neurofilaments in nerve cells of the embryos. And this gives you more information about the intermediate filaments. So the fundamental subunit of intermediate filaments are dimers, and intermediate filament proteins are fibrous rather than globular proteins. And this is some more information about them. So these proteins, they have homologous central rod-like domain of 300 to 318 amino acids, and flanking the central helical domain are N and C terminal domains that differ greatly among the intermediate filament proteins. The basic structural unit basically consists of filament polypeptides intertwined into coiled coil. So you can see here, so basic structure is a dimer like this. Okay, so it consists of two intermediate filament polypeptides intertwined into a coiled coil. So this is one dimer. Okay, sorry. The two polypeptides are aligned in parallel, as we just saw, and two such dimers align laterally to form a tetrameric protofilament. So these are two dimers, and they can form a tetrameric protofilament. And the protofilaments overlap to, so they form the two dimers, they form the tetrameric protofilament, and the protofilaments overlap to build up a filamentous structure about 8 protofilaments thick. So you can see here that this is the tetrameric protofilament, and then around 4, sorry, how many, once again, then the protofilaments actually overlap to build up a filamentous structure which is about 8 protofilaments thick. So you can see that these tetrameric protofilaments, they can overlap like this, and they can form filamentous structure about 8 protofilaments thick. So they can form the structure like this, and it has how many? 8 protofilaments. So this is one intermediate filament. So first of all is a dimer. The dimers form tetrameric protofilaments, and then these protofilaments, they overlap and they form the structure. They form the structures like this, which has 8 protofilaments, and this is one. It's called the intermediate filament. So protofilaments overlap to build a filamentous structure which is about 8 protofilaments thick. And something very important to know about intermediate filaments is that they have a tension bearing role, and they are less susceptible to chemical attack than the microtubules and microfilaments. So that's all about this chapter. So basically this chapter is all information, so you can just it's easy to understand. It's basically information. So please let me know if you have any questions.